What is a dinoflagellate? Dinoflagellates are unicellular protists that inhabit ocean surfaces. They are deemed planktonic, and the majority are marine plankton. Most are microscopic and tend to range from 15 to 40 microns in diameter, but the largest dinoflagellates can be about 2 millimeters in diameter. The observation of dinoflagellates began in 1542 with Spanish explorer Alvar Núñez Cabeza de Vaca's descriptions of algae blooms that emitted harmful toxins into the ocean, affecting the marine life around them. They were first officially examined in 1773 by Danish naturalist Otto Friedrich Müller, who named them from the Greek dinos, meaning whirling, and the Latin flagellum, meaning scourge. This is a diagram of the sectioned theccate of a motile dinoflagellate. If an organism is motile, that means they have two flagella, which are movable protein strands that help propel the eukaryotic organisms through the water. There's the nucleus, chloroplast, Golgi body, mitochondria, and then the flagellum. Theccate dinoflagellates have internal skeletons made up of cellulose-like plates. Some dinoflagellates called naked cells lack the theccate that would form the rigid plates of the membrane.
Now we're going to be talking about red tides. A red tide is a term that describes a discoloration of water caused by a bloom of red dinoflagellates. Red tides are deadly to marine life. And now I'm going to draw some fish that have a first-hand account of these deadly dinoflagellates, which is sad. Scientists prefer to reference these blooms as harmful algal blooms. Red tides occur when dinoflagellate populations increase to a very large number. So I will proceed to draw two dinoflagellates that look like people, and my brackets kind of suck, but that's okay. So scientists have not been able to fully understand the environmental conditions in which these blooms occur. However, they do believe that the blooms are a product of specific nutrient and hydrographic conditions. Red tides are entirely made up of one species of dinoflagellates that grows quickly. Red tides are known for their emission of toxins, but not all red tides emit them. Interesting. So here's a crab, and he is representing the shellfish. And when the dinoflagellates are consumed by shellfish, they accumulate a high enough concentration of chemicals in their tissue that cause brain damage to humans and animals that consume these shellfish. And the crab is just worried because he ate a dinoflagellate and could give people brain damage. This poisoning is called amnesic shellfish poisoning and is a domoic acid and attacks the brain. There are various types of toxins released by dinoflagellates and can affect neural synapses by interfering with the movement of ions across cell membranes.
So what have we discussed? So far we've discussed the properties and habitual tendencies of dinoflagellates, and we have discussed red tides and their environmental effects. So what causes the emission of light? Since dinoflagellates, that's a bad drawing of dinoflagellates, are microscopic, the bioluminescence can only be observed with a high concentration of them. So chemically, the light is produced through an oxidation reaction between luciferin and oxygen. So luciferin is a complex molecule, as that can be seen by my drawing of the molecule, and it does not fall under any of the common classifications of compounds, but it is similar to chlorophyll that is found in plants. And because it's luciferin, I thought I'd draw a devil, which was weird. Okay, let's move on. So as I said, it's similar to chlorophyll that is found in plants. So in this reaction, the luciferin is oxidized by the addition of oxygen molecules with the assistance of the luciferase enzyme. The reaction of this production of light is a result of the addition and the emission of energy within the atoms involved. And this bioluminescence is characterized as cold light due to the lack of heat involved in this emission. The reaction of the production of light is a result of the addition and the emission of energy within the atoms involved. And that is my drawing of an atom involved. So how is this carried out? So the absorption of the energy allows the electrons to move up in energy level. So here's my drawing. I screwed up. I meant to do a dot, but I accidentally drew electrons in the center of the nucleus, but they're protons. And they're also neutrons. So I'm using Bohr's method to show the change in energy. So the electron gains energy, so it can move up an energy level. And the atom is deemed excited. So the emission of light is the electron's release of energy. Here's once again the drawing of the electron shell, and I can't draw circles. So this is currently the excited electron. And now to move back to its ground state, it moves back down to its original energy level. So the energy released is light. Thank you for watching the video, and thank you for enjoying the Dinoflagellates. Thank you all, and that's a Dinoflagellate.
Yep, I drew that.
Mary Margaret did draw that. I don't know who drew that. Okay.
I drew that too.
So, Dinoflagellates and the emission of light. And oh, here's a joke. Why does a burger yield less energy than steak? Because it's in its ground state. Ah.
And there's a heart with a smiley face, and then I had to draw some eyebrows. And
I am funnier than Mary Margaret.
What? I didn't even know you wrote that.
And that's Babe Lincoln.
Okay. Thank you all.
Thank you.
Woo.
Oh.